I'm very honored to introduce Dr. Johanna Melke, who is a senior scientist at Mosa as our keynote speaker for this session. Johanna's got her degrees in biotechnology and molecular biology and her PhD in biomedical engineering from Eindhoven University of Technology. She's been a member of the FAT team at Mosa since 2019 and will showcase the multifaceted approaches that are needed to get to diverse tissue types for cultured meat, including fat. What we're going to see throughout this session is that it's not just cell selection or media or the cell microenvironment that we need to yield diverse tissues, but actually a combination of all of these things. And as with so many aspects of cultured meat, we're really building on foundations from regenerative medicine and biomedical sciences. And I'm really excited for Johanna to showcase how we've grown from these foundations into where we are today with her work from the very, very prolific FAT team at Mosa. So pleasure to welcome Johanna.
Thank you. Thank you very much. Thank you for this really nice introduction, and thank you for the invitation to speak here. I'm honored to talk on behalf of the FAT team, not only the FAT team, also its extended and honorary members. There are more people beyond the FAT team that are working on FAT and contribute greatly and whose work we're really benefiting off. And I just wanted to take an opportunity to showcase the progress that we have made in the last years and also show some very crucial steps that are really important to get us to where we are today. So let's get right into it, starting with the mandatory introductory slide, just a quick overview of the process, which consists of cell selection, cell proliferation, and tissue maturation. At Mosa we want to produce a hamburger made from beef, so of course we're starting out with a cow, and there we're taking tissue. We're not working with embryonic stem cells or iPSCs, also not really. We're focusing more on cells isolated from tissue. These cells get sorted and then go into a large proliferation cycle where we're producing billions and billions of cells. And eventually, once we have enough cells, they go into differentiation. We let the tissue mature. That can be done as aggregates on scaffolds in a hydrogel. There are different ways to do that. And eventually, once the tissue is mature, it's going into processing to turn it into an edible product. So let's start right at the beginning and start at the cells. So when I started, which was like 4 years ago, working mostly with cells isolated from fat tissue, which is kind of the logic choice, right? Like if you want to make fat, you take stem cells from fat. And that's what we use to produce our fat tissue. But there was also Richard. Back then, he was a very young PhD. Now he's almost finished. And he was working with these fibro-adipogenic precursor cells, which back then we called population 4 because we had not really any clue what these kind of cells were. And he was very convinced that these cells were a really good alternative and they had potential for fat tissue engineering. And the advantage of these cells is that they are also found in muscle tissue. So if you're already isolating satellite cells for muscle tissue production, you can find these cells in the same tissue and you kind of isolate both in one go. You just digest your tissue, all the cells come out, you can fax them, sort them, and then you get basically these FAPs, these fibro-adipogenic precursor cells and satellite cells. And he showed very nicely, let me see if this pointer's working, oh yeah, it does, that these FAPs are not able to differentiate into muscle, unlike the satellite cells. They can do it, of course, but these FAPs are quite good at differentiating along the adipogenic lineage, which the satellite cells cannot do in turn. So that was already quite promising, and he continued his research, and we could show that these cells can accumulate quite a lot of fat, also in 3D. So here we see the progress over 4 weeks, where the cells are in an alginate gel. So this is a 3-dimensional tissue, and they can produce quite a considerable amount of fat. And you can also see with lipidomic analysis that the fatty acid profile changes quite a lot as the cells mature over time, and it gets kind of close to what we see in fat and muscle. And also from the looks of it, of the entire tissue, we can see that the empty gel, of course, it's just a translucent alginate gel, but if you put in cells and you differentiate them, then it turns very like yellowish, so fat accumulates, and it looks a bit more similar to what we see in real fat. And the cells at the beginning are, of course, quite small on day 0, but then over time, as they mature, they get really large, and you can see maybe these like tiny fat bubbles that are trying to press through the membrane. So also there we see that the cells are getting closer to the tissue that we're actually aiming for. So yeah, these FAPs suddenly became really interesting. And later on, we also saw that these FAPs are doing quite well in long-term proliferation. So also under serum-free conditions, we could see that you can go well beyond 30 PDs. I think, we have cracked these numbers already like a long time ago. So long-term proliferation, of course, is very important if you want to upscale and you want to get to production-relevant levels. So proliferation, basically check, but then we also wanted to look at differentiation. Can the cell long-term proliferate? Can they also differentiate after? And there we saw that not only do they surpass our standard cells that we isolate from fat tissue, so adipose-derived mesenchymal stem cells, or SVCs, because they're coming from the stromal vascular fraction. So they lost their stemness or differentiation capacity relatively quickly. This was after proliferation on microcarriers, so this was done in spinner flasks and then differentiated in 3D. And these FAPs, they kept their differentiation potential quite long. You can see here all these little yellow bubbles, which are all the fat that they accumulated. So yeah, they surpassed our standard cell, and they also were able to retain their differentiation capacity for quite some population doublings. So here we did a couple of isolation screenings, which were mostly done by Celine and Federica. So they screened dozens of different isolations, and these cells were proliferated also in spinners on microcarriers, harvested, reseeded, and every time we did a little differentiation to see how the cells would progress. And of course, there's always a little bit of variation. We have isolations that are better than others. Isolation E here, for example, is not very great. Also drops in stemness very quickly. Isolation C, we see a little drop towards the end. So there's always donor variations, which is just something that you always have to deal with if you're working with primary cells. And that's also the reason why we're doing extensive testing of all these different isolations, because it would be a shame if we give isolation E, for example, to the production team. They start a 1,000-liter bioreactor only to notice after a couple of weeks that there's no fat accumulated. So stemness is always a challenge, and if you're more interested in differentiation versus age of FAPs, Richard is going to give a talk in the same session, so stay tuned. But yeah, basically we made the switch to FAPs. Wasn't really hard, because these cells are really impressive and they have such a great potential for fat tissue engineering. So the next step was the differentiation. So there, one very important step was the development of a serum-free differentiation media, and that's work that Radha did, who is also a PhD and also about to finish up. And she basically did two things. So once she looked at replacing FBS, so she developed this serum-free differentiation media, and not only did the cells differentiate, they differentiated better than in the FBS version. So that was already very interesting, and we already saw like a great increase between the lipid accumulation. And on top of that, she also reduced the medium. So before we had rosiglitazone, insulin, dexamethasone, and IBMX in there. She kicked out the dexamethasone and the IBMX. Of course, less compounds is always cheaper, but also it's about food safety. Not all compounds can go into medium if you're producing food. So these two compounds went out, and there she also saw in FBS but also in the serum-free medium that there's more differentiation happening. So we were left with rosiclitazone and insulin. Rosiclitazone, of course, also a compound that you don't want to have in your medium. It got replaced. Even the replacement is now being replaced, so there's like constant movement in the differentiation protocol. But that was a very important step, developing this serum-free media. And on top of that, she also showed that it works across species. So she tested this not only in beef, but also in pork and also in sheep. They react differently to these like differences between FBS and serum-free medium, but serum-free medium worked in all of them. So it kind of gives us, kind of the freedom to also work on other species, not really something that we're actively pursuing, but we could if we wanted to. So that was a very important step. So we had kind of the base for the medium. And the next step was something that Sophie already presented before. So waste reduction was really, really important. So she showed very nicely that ammonia is really detrimental to differentiation. And that figure we have already seen before. If you spike in ammonia, the differentiation is really reduced. And so she came up with this medium that instead of glucose and glutamax would contain galactose and pyruvate. And from there, we could see that not only did the cells keep differentiating, they actually differentiated way more. So we got like approximately twice, a little bit more than twice as much fat. And the cells would not produce any waste. And that was a very important step, because from then on, we could basically modify how much medium we feed the cells. So there were a lot of iterations, reduce the media volume that the cells get set up in, but also reducing media changes, and through like lots of testing, what kind of compounds the cells need, we managed to not do media changes at all anymore. So we can see here that there are good isolations, there are bad isolations. If you do not change any media, that's not great, that's going to reduce differentiation. Not because there's any waste accumulated or like negative proteins being secreted, just because the cells are very likely running out of nutrients. But if you put in the nutrients that they're consuming, you can gain back this differentiation potential. In this case, we just added an amino acid cocktail. So no media changes, just spiked in amino acids that the cells are consuming. And we ended up with an even better differentiation than our control condition. Maybe because the cells are producing factors, could be short-lived molecules, could be like entire proteins. So they're not getting depleted by media changes. And have a positive effect on differentiation. And just by doing that, we could reduce our media volume by 90%, which of course is like a really good cost reduction. And there we didn't even start looking at other ways, like going from pharma grade to food grade, going from proteins or from like entire growth factors to just peptides. So there are a lot of different ways how to reduce the media even further, get the cost down, and do more high throughput screenings to push the cost to like a really minimum. And talking a little bit about high throughput screening, I just wanted to throw in that slide to show the different workflows that we have at the moment in the team. So there of course, high throughput screenings available, which we're doing in 96 or 384 well plates. It can be done in 2D. We also have ways of putting cells into like a thin gel, like an alginate gel that is thick enough that the cells are kind of in a 3D environment, but thin enough that the high content analyzer can analyze this. We're also looking a little bit more lately into aggregates, which have quite some advantages. You don't need any extra gel or extra scaffold. So that's something that has come up a little bit more often lately. And if you're interested in aggregates, please check out Vicky's poster. She has done some work on aggregates, if you're interested in that. And then, of course, our kind of main deal is still the alginate gel, where we just incorporate the cells into the alginate, and we can do different shapes. The initial shape was always a spaghetti, because that's what you get if you extrude it from a syringe or from your pipette. But it has some disadvantages. If you have anything that has an impeller, the spaghetti likes to get tangled up in it, and it just forms clumps. So we also started to make a different shape, which are just beads. And that is much easier if you have a spinner or a bioreactor, and the beads can just freely float in there, and they're not getting entangled. So these beads or spaghetti have also undergone some optimization, and there I would like to show some work that Sebastiano has been doing, where he just played around with different seeding densities. Of course, for a production, it's much easier to run a liter of 120 million cells per ml instead of processing 4 liters of 30 million. So that's just practicality. And we just wanted to see if increasing the cell content would be detrimental, if there's issues with diffusion, if we would see a drop in differentiation, that the cells don't have enough space. But it turns out adding more cells is quite a positive thing. So when we doubled the cell number and then doubled it again, we saw that the amount of fat that the cells are accumulating is going up. So that was a quite positive outcome. Plus, on top of that, if you want to get rid of the gel, so these cells are still in alginate, and you would want to get rid of it, maybe for product development purposes, then these cells that are in a very high density, they keep their shape, probably because during culture they start to reach out to each other, they start to connect. So once you remove the alginate through citrate addition, which is pulling out calcium, so the calcium from the alginate goes out, and the gel falls apart. In the very low concentration, seeding concentration, the cells will just disperse and go into single cells. But if you have a very dense tissue, everything stays the same. The alginate disappears, but the cells don't really mind. They keep the shape. So then Sebastiano also looked at another way of optimizing this culture, which looked a little bit more into biomechanics. And biomechanics and tissue engineered fat is not really something that people straight away connect. It's usually more like mechanobiology and bones or cartilage, and shear is important, and you want to stimulate your cells. But fat is quite the opposite, of course. It just wants to be left in peace and not being steered too much and shaken too much. So he did a really nice experiment where he just cultured the cells or the fat in spinner flasks at different speeds. So 23 RPM seems like such a random number, but it's basically the minimum you need to lift the tissue off. Otherwise, it just sits at the bottom. There's not enough mass transfer. They just starve basically and suffocate. So 23 is the minimum that you can get everything moving. And then he went all the way up to 120 RPM. And you can see nicely how the differentiation drops quite a lot. So the best, of course, was the slowest. So 23 RPM has quite a good fat content, and then it slowly goes down. And you can also see here, that's how it looked like in real life. 23 rounds per minute, so very gentle steering. The cells, the whole tissue is floating at the top. Here it's kind of hovering somewhere in the middle, and then it's just sitting at the very bottom, all set and non-differentiated. So that's kind of one important fact, that you don't just throw your cells into a bioreactor, steer them, and then just hope for the best. Also biomechanics and shear rates that are the cells are exposed to are quite important and something to think about. And basically now the tissue is perfect in a way, right? We have the perfect cells, we have really good medium, we know how many cells to seed, we also know how to culture it very gently. So now the next step is, of course, product development, which is something that Francesco and Sebastiano have been taking care of, and it's no coincidence that it's two Italians. They just know the food. So they took on this challenge to make sure that looks and shape and consistency is up to what consumers would expect and demand. And they also investigated like how we would incorporate the cultivated meat, what amounts would go on it. And like one of the first challenges was if you want to make a hybrid burger, to get a base. So first they basically developed this plant-based burger, tried out a lot of recipes. So we would have something where the cultured meat or cultured fat could go in eventually, and they managed to do that with like a lot of different iterations, and they came up with a burger that in itself is savory and salty and caramelized, has a good texture, the color shifts, it has in-pan aroma development, so it was a good base. And to this burger, they eventually added the cultured meat, so cultured fat, cultured muscle, and that just made it better, of course, because then, of course, you had this fatty and meaty aroma. There's the smell of raw meat, and the whole organoleptic properties really improved with this burger. And if you're interested more in product development, Sebastiano's going to give a talk on that, I think, in November in Las Vegas, if I'm right. So he's gonna expand a little bit more on this whole product development. So in conclusion, kind of take-home messages. I hope you kind of agree that FAPs are incredible cells that are extremely suitable for a scalable cultured meat production process. They can be proliferated to really high numbers. They can go to really impressive population doublings and maintain the adipogenic potential. They can mature into like 3D fat, and they even outperform our classic cells that we used to isolate from fat tissue itself. The next step was, of course, the serum-free differentiation, META, which outperforms FBS by a lot, even across different species. And the next step of substituting glucose and glutamine with galactose and pyruvate really promoted FAP differentiation so that we could achieve even more mature adipocytes and make the process more scalable, cheaper, more efficient. And then Seb basically showed that higher seeding cell densities are promoting differentiation. And at the same time, if you lower the shear to as little as you can, basically, that can also increase lipid accumulation. So also shear rates, very important. Biomechanics should not be neglected, even if fat seems to be such a lazy tissue that doesn't keep up with things like cartilage or muscle or even bones. And then lastly, the addition of cultured muscle and fat really improved the organoleptic properties of plant-based burgers. So it is an option to go for a hybrid burger in the end, which is cheaper and easier to produce in the end. So, of course, there's like a lot of people that are involved, not only the infamous FAT team, but also all the people from other teams who contributed to this work, people who supplied us with cells, who supplied us with medium, ideas, feedback, like data acquisition and data sorting. Like this was really a very, very big team effort. And that's the only way how we could achieve so much in such a short time, even though 4 years seems like a really long time. But time passes very quickly, and I would just like to show where we were like a couple of years ago, where just some sad cells sitting in a well plate, differentiated, if we really asked them very nicely, and then within 4 years we made it to a real 3D tissue, which is swimming on top of the medium, and that can actually go into a burger eventually. So if there are any questions, I'm happy to answer. Oh, many questions. So much. That's good.
The box for questions is over there.
Thank you for the wonderful talk. Really, really interesting. And that picture on the bottom right is just beautiful. So I'm curious, when you do the seeding density difference, you see sort of this split into like, it looks like sort of a bimodal where you have some cells that are accumulating a lot of lipid droplets and then some cells which are accumulating fewer at the higher cell seeding densities. And I'm just wondering what your thoughts are as to sort of what's happening there.
So I think once you're reaching these really high cell seeding densities, you're kind of crossing the line towards also an aggregate. And I think the cells are very much communicating with each other. So you can have this kind of non-homogeneous distribution where you see like a part that is differentiating really well, and then like the opposite side is not differentiating at all. And we're still wondering like what these cells tell each other, like why, for sure something must happen, right? Like something that shouldn't be in the media. It must be some communication that is happening like on a really short scale, or they're like directly communicating with each other. So that's something that we're also trying to understand. And we see that more often, that the more cells you put in, the more you see kind of these waves of differentiation, which is really interesting. We're also trying to figure that out.
We have time for one more question. Oh man, so many. I'm sorry, guys.
Crazy stress.
Maybe let's keep it in this direction.
You're the lucky one. Oh.
Okay, thank you for your presentation. What is the fat composition of the cultured cells compared to the normal fat? Fatty acid composition.
It's very close. It's not the same, but we're getting close. We have a lipidomic analysis, and we see that over time as the cells mature. So we only went up for lipidomics until 28 days. And we see that the lipid profile changes a lot, which is, of course, at the beginning, the cells have no fat. So like small changes are reflected like in a big percentage, basically. But then over time, when the cells mature and they get really big droplets, then the fatty acid profile gets really close to what we find in muscle tissue or in fat tissue.
Or can you manipulate the fatty acid composition using the substrate or the GM?
You can add, for example, fatty acids to the medium, which is not something that we're doing. But yeah, you can add different fatty acid cocktails, which doesn't mean that the exact percentage is going to translate into what you see in the lipid droplet in the end. But that's definitely something that is possible.
Cool. Thanks so much, guys. I think we need to go to the next speaker, but thank you so much, Johanna, for your presentation and everyone for your questions.
Thank you.